So I want to talk more about the experimental basis of what the textbook tells us that HSP70 does. I'm going to focus on HSP70 alone, all right? So, because we can figure out a lot of the properties of HSP70 by the techniques of pulse chase analysis that we've talked about before, coupled with immunoprecipitation. So it's a really great practice of what we've talked about before. So, so what I'm going to do is give you an experimental setup, and then I'm going to break it out into smaller tidbits to focus on unique aspects of this very complex experiment to see what conclusions we can reach. All right? But the general idea is that we are using bacteria, E. coli, that also contains its own version of HSP70. It's not called HSP70, but it is the same protein, so I'm calling it HSP70. E. coli HSP70. All right? And and you can grow them a lot very nicely and cheaply in cultures. So what these researchers did was to grow E. coli that contain HSP70 or a mutant form of E. coli that lacked that HSP70, right? And what they did was to see what happened with pulse chase and immunoprecipitation. They had an antibody that they claimed was specific to HSP70. That claim we need to validate, of course. Right? How do we know that they're doing the experiment correctly and that the reagents are any good? So all of that is addressed by the use of controls. So we're going to go through their experimental plan and controls. Right? So I just told you that the chaperones are really important, and without them, cells are going to essentially die. How can you have an E. coli that does not have HSP70? Because it's not actually totally true. So just note of clarification. Bacteria have multiple different types of HSP70s. So this is one of them that we're looking at, but the cell can then limp along with the other HSP70 that is hanging around, all right? But because it's different enough, our antibodies won't bind to that, we are not gonna worry about it, all right? But that's how these cells are alive, all right? Just to explain that. So here's the idea. So we can take, just to recall the process of pulse chase and immunoprecipitation. So you can take cells, give them a short pulse of a radioactively labeled amino acid, in this case methionine, S35 methionine, and then the cells will gobble that up and use that in the production of proteins during that pulse. Proteins that were made before are not radioactive, and then proteins that are synthesized after the pulse, when we give the cells, wash off the radioactivity and give them non-radioactive amino acids, those are not going to be radioactive either. Only proteins made in that pulse are going to be radioactive. So at different times after the pulse, we can take a collection of cells and break them open. So important things to consider. While you are not opening the cells are alive, and all the normal mechanisms are going to occur inside the cytoplasm of the living cell. Right? So things are going to be happening. When you open the cells, you're going to stop that, all right? And you are going to open up the cells into a surrounding buffer. And now this is the important part too. The surrounding buffer is going to be asking a variety of things. So we are going to be using different types of buffers in each of these experiments. All right? This experiment shows only the pulse. All right? What happens after the pulse? There's no chase here. The chase is coming up a little bit later. All right? The figure of the chase. So we are in this first gel. We give them the 1 minute pulse, lyse them in different buffers, and then we use an antibody that is supposed to be against HSP70. So to review that, again, we use beads that have our antibodies on their surface. We reach into the lysate of those broken up cells to grab the HSP70, supposedly, specifically, and then wash off the beads from any non-specific binding. We put hot SDS and beta-mercaptoethanol loaded on a polyacrylamide gel, run that on the gel, then we dry the gel, and because we want to look at radioactive proteins, we put a film. I didn't bring my film again. We put an X-ray film to be exposed to that radioactive gel, and this is the image of the X-ray film. This is not a Western blot. This is an X-ray film. All right. Good. Questions? Okay, so what are all of these buffers? So here's all the composition of all this experiment. This is a lot to go through, so let's just break it up into small parts, all right? So first question, and I'm going to set this up on Instafor as well, but let's just talk about the experiment, all right? So in this experiment, we are running lysates that were done in the absence of ATP added to the buffer. So just a nice buffer that maybe contains a little bit of salt, pH buffer that keeps the pH constant, and that's it. In lane 2, we add salt and buffer to the solution, but we also add extra ATP. All right? So that's in lane 2, in the presence of added ATP. In lane 3, we do in the added presence of a smile detergent called Triton X100. Not strong enough like SDS to the nature proteins, but it is able to act as a detergent, has a hydrophobic part. And then lane 4 has the mutant bacteria that doesn't have HSP70. Right? All of these were, again, pulse for 5, 15 minutes, and then right after that, we break them up in different buffers, do the immunoprecipitation, and load that on the gel, do the X-ray film, all right? And notice that we have a band at about 70 kilodaltons that it's marked there as HSP70 with the arrow. So the first question I'm asking you is, okay, so we see HSP70 right there. Why? Might seem like an odd question, but think about it. Why do we see HSP70 right there? Okay, so the majority of you are wrong. And there's 3 answers that are favored among all of you. So find somebody with a different answer than you and have a good chat about it. It's very important that you remember everything that has been done. You have taken the immunoprecipitate, you have boiled it with SDS, remember what that does? You run it on an SDS acrylamide gel. What does that do? And then you expose that to an X-ray film. All of that is important to answer this question. Okay.
What do you guys put in?
One of the answers. Three answers.
No, I mean, I get it.
What do you guys have?
Okay. So, obviously, we think it is as what I was saying. Right now, because I feel like this could also be the same question that last time when it was just wait, no.
All right, give me a vote again, please.
So basically, if it came with HSP70, when you nature it, you can separate from your proteins as opposed to different weights. So you see like down here.
So it looks like I have a mano a mano between D and E. So last chance to vote. There's a few B's, but majority of you 50-50 on D and E. So let's hear, well, let's just have a discussion here. All right, I'll close it now. So, picture what's happening, all right? So the cell has just made radioactive proteins, right? And let's just say that they are bound by HSP70, all right? So there's a protein of 50 kilodaltons, right? That is bound by HSP70, which is 70 kilodaltons, all right? We open the cell, right? And now we grab with our tweezers, with our antibodies, we grab HSP70. So we are taking HSP70 out and we load it on the gel, right? So therefore we should see it on the gel. But anything that is bound to HSP70 should come out as a co-immunoprecipitate as well, all right? Why do you see any proteins on this film, X-ray film? Because they're radioactive, right? So the fact that you see HSP70 there, it means it's radioactive, right? If it wasn't radioactive, you would not see it. So the fact that it's radioactive means that it was made radioactive. And when was it made radioactive? It was made radioactive during that pulse. So when you are immunoprecipitating, when you're grabbing HSP70, the cell had a whole bunch of old HSP70 that was not radioactive. Are you grabbing that? Yes, you are grabbing it, right? Because the antibody doesn't distinguish between radioactive and non-radioactive. So the antibody grabs all HSP70, not radioactive and radioactive. And you're running that on the gel. And all of that is migrating on the gel. So right there, on that HSP70 band weight, right, there's a whole bunch of HSP70 that is there. It's just not visible because it's not radioactive. We're only looking at those few HSP70 molecules that are radioactive because they happen to have been made at that moment. All right? So that's why we see HSP70.